Hello everyone, welcome back to BMI5730. Today we will continue with proteomics. We will talk about tools, databases, and pipelines used in proteome informatics. In previous lectures, we have talked about basics in proteomics applications. We talked about protein identification and quantification. For protein identification, an important technique is called peptide mass fingerprinting, PMS, also known as protein fingerprinting. This technique for protein identification was developed in 1993. It has the following steps. First, generate a query peptide peak list. The unknown protein of interest is first cleaved into small peptides, for example by trypsin digestion, whose absolute masses can be accurately measured with a mass spectrometer. Second, prepare the protein sequence database. The peptide masses are compared to either a database containing known peptide sequences or theoretical peptide sequences computationally translated from a genome. Third, search the database to find the best match. The masses of the peptides of the unknown protein are compared to the theoretical peptide masses in the database. The results are statistically analyzed to find the best match. Finally, protein identification. Evidence of the existence of peptides is integrated to signal the existence of proteins where the peptides came from. We have tools and databases that can help us complete these steps. For example, Mascot is a software that uses mass spectrometry data to identify proteins from peptide sequence databases. In proteomics, we also call such a tool a search engine, because it searches databases to find the best matches. Mascot is a widely used software using a probabilistic scoring algorithm for protein identification. It looks for the highest scoring set of peptide mass matches from the database for the query proteins. Peptide mass fingerprinting is a technique, while peptide mass fingerprint is a peak list. Mass spectrometers output raw data representing peak lists. Different mass spectrometers may generate different data formats. Some of the tools can process multiple types of data. Take Mascot, for example. It can automatically recognize several formats. There are also tools that can convert some data format to another. As described on Mascot website, for a peptide mass fingerprint, the file should contain a list of peptide mass values, one per line, optionally followed by a white space and peak area or intensity value. The peak list formats of a wide range of instrument data systems are directly compatible with these requirements. For protein quantification, there are different experimental techniques that use different quantification approaches. SciLac, iTrack, and iCAD can generate relative quantification. For example, using SciLac, you can generate a ratio of the abundances of proteins from two different samples. From some targeted mass spectrometry methods, such as MRM, you can get absolute quantification. There are also semi-quantitative, label-free methods where you can get spectral counts. The spectral count is defined as the total number of spectra identified for a protein. It is not absolute quantification, but very practical and well accepted. We will not go into detail about the chemistry of SciLac and iTrack. Please feel free to search Wikipedia after the class for a brief introduction. SciLac stands for Stable Isotope Labeling of Amino Acid in Cell Culture. Basically, you can label the same peptide from different samples with different stable isotopes. The control sample can be free of isotope labeling. Just use well-typed amino acids. In the resultant peak list, you can have the pairwise comparison of the same peptide from two different samples. iTrack stands for isobaric tags for relative and absolute quantification. It is another method with labeling. It can label up to 4 samples in the same experiments. Now we move on to protein sequence databases. We need them in conventional protein identification. There are a bunch of online databases available. ExPaSy, the expert protein analysis system, is the godfather of all bioinformatics websites. UniProt KB, SwissProt, and Trumbo, we use them often in proteomics. PDB, the protein data bank, has protein information, including protein structures. In addition, ProCite, a database of domains, contains information on protein families and functional sites. UniProt is a comprehensive, high-quality resource of protein sequences and functional information. It contains 4 flavors of databases. We are most interested in UniProt KB. UniProt KB, the knowledge base, contains SwissProt and Trumbo components. This corresponds to manually curated and automatically annotated entries, respectively. Trumbo contains automatically annotated, unreviewed data. It has redundancy. SwissProt contains high-quality manual annotation. Its data is reviewed and non-redundant. One more page as a note for you to tell the difference between this SwissProt and Trumbo. The redundancy in Trumbo is due to various reasons, including false predictions from algorithms and sequence errors, etc. So you know why its quality is not as high as SwissProt's. The main information added to a SwissProt entry includes sequence, variants, nomenclature, sequence features such as domains and motifs, literature annotations, ontologies, and references. It is very comprehensive. UniProt provides cross-references to a large number of other resources and can serve as a useful portal when you first begin to investigate a specific protein. You can download the sequence database from the UniProt website. Pay attention to which version you want to download: reviewed SwissProt or unreviewed Trumbo. You may download the data format as required by the software you're going to use, say SwissProt FASTER file. On UniProt website, you may search a protein by name, such as CD274. The website also supports peptide search. You may go to the website and play with different search functions. Interpro is another web tool that provides functional analysis of proteins by classifying them into families and predicting domains and important sites. You just need to submit the protein sequence of interest. You may search TP53's CDS sequence and see what you can get. Play with the tools. Now we will introduce some databases of mass spectrometry raw data. Besides downloading data from the existing works of other researchers, you may also deposit your own data there. Peptide Atlas. It is maintained by Seattle Proteome Center. It is an accessible compendium of peptides identified in a large set of tandem mass spectrometry proteomics experiments. Mass spectrometry data are collected from human, mouse, yeast, and several other organisms. All results of sequence and spectral library searching are subsequently processed through the transproteomic pipeline, that is TPP. You can search for protein sequences and protein. You can download the raw data, search results, and full builds. This is the homepage screenshot of Peptide Atlas. Here's another database. NIST Mass Spectrometry Data Center holds a peptide library at this site. Another one, Massive, the Mass Spectrometry Interactive Virtual Environment, also has a large proteome data repository. It is hosted by UC San Diego. Last but not least, for people interested in cancer proteomics, CPTech Consortium hosts the proteomics data for the TCGA project. You may check the data platforms and data types they have. Okay, we talked about databases. Now let's talk about the mass spec data formats. There are multiple formats. This is a screenshot from Mascot website. Mascot can support more than 10 formats. Let me introduce several to you. MZ data. HUPO Proteomics Standard Initiative, PSI, developed this XML format meant to unify the existing formats into a standard format for reporting peak list information called MZ data. Essentially, MZ data provided standard means to represent peak data from a mass spectrometry experiment and establish fundamental parameters necessary for proper interpretation of the spectra, such as negative or positive ion spectra and other basic information. MZXML. MZXML was developed by the Seattle Proteome Center at the Institute for Systems Biology. This data format is similar in its minimal requirement for the representation of MS data, but with several key differences which make it the preferred file format for MS data. MZML, resultant of the HUPO Proteomics Standards Initiative's continuing progress towards standardization of proteomics data, specifically mass spectrometry data, led to the development of a new unifying MS data format called MZML. How does an MZXML file look like? Well, it is still an XML file, so it follows the general structure of XML. For example, the scan of peaks is located in a chunk between the start tag scan and the end tag /scan. Well, Mascot also provides generic format. We call it Mascot generic format. It contains the same information as peak list, just that it converts XML format into a plain text format. In Mascot generic format, in the case of peptide mass fingerprint, each query is just a single peptide M/Z value with an optional second value for peak area or intensity. The example I highlighted here has two columns. The first column are M/Z values of the peaks, and the second column are the intensity of the peaks. You may have known that for a peak list, as shown in the window, the x-axis is always M/Z value, and here the y-axis is relative intensity. Okay, after talking about databases, we will talk about search engines. There are many options, including Mascot. Here I provide a list, just for your information. Please feel free to check those websites and get to know more. There are also multiple integrated pipelines. Using them, you can perform protein identification and quantification and further downstream analysis. Example, Scaffold. It has both commercial and free versions. We will try to analyze some example data using the free version Scaffold. MaxQuant followed with Perseus. This is another pipeline developed by Max Planck Institute. Last but not least, TPP, the Transproteomic Pipeline, developed by Seattle Proteome Center. Acknowledgements. Thanks to all the tools, databases, and pipelines we can use to perform comprehensive proteomics data analysis. The advances in bioinformatics have made our tasks of data analysis more beautiful and feasible. Thanks for watching. See you soon.